Welcome to the Job Market Insights, a weekly webinar series by UCSC Silicon Valley Extension. So let's just go ahead and dive right in. One of the common questions we received was, how safe will the vaccine be?
So I made an appendix of some anticipated questions. So I think when the vaccine finally is approved, it will be pretty safe. So this is one of the questions that kind of comes up, where can I find out about the safety of a vaccine? So one of the things you can go to once a vaccine is approved by the FDA, the package insert will be posted at the company, the website of the company who developed the vaccine. So this is the influenza vaccine. It's the Fluvarex quad vaccine. So it's the quadrivalent vaccine for flu for the 2020-2021 season. But GlaxoSmithKline has actually manufactured this vaccine for probably 15 years. They had to make some changes as a result of different flu viruses circulating in any given year. But in the package insert, there's tons of information about the safety of the vaccine. So this is page 1 of the insert. So here's kind of a high-level summary of the adverse reactions associated with the vaccine. Then when you go down the rest of the package insert, you want to focus on information in section 6, and I just took one table. So this goes to show the adverse reactions or side effects of the vaccines, the local systemic, the severity of those vaccines in patients who receive the vaccine versus patients in placebo. So this is from the age group 18 to 64, 65 years older. So there's tons of information on vaccine safety. So this is one place you could look and hopefully be satisfied with the results. But the other thing is companies publish in journals. People who are not associated with the companies will be reporting on drug safety. So there's tons of different places where you can go on the internet, in the scientific literature, to the company itself, to the FDA website on these, to actually find out, basic questions on, relating to how safe the vaccine is. So just like drugs, all vaccines have side effects. So, you do have to be comfortable with the reality of, experiencing a side effect. But for the most part, the severity of the side effects are, nothing too significant. They will pass in a few days and you'll be on your way. And if the vaccine is effective, then you'll be protected from SARS, going forward based on, whatever the findings are with how long the immunity lasts to that vaccine. So to me, the answer to the question is, I think they can be pretty safe. The FDA will not cut corners. NIH will not cut corners. You can't release vaccines to the community if they're really substantially not safe. What I think I would be most concerned about is, will it be immunogenic enough, given the history of vaccine development? But as I say, first vaccine comes out, there's probably going to be a dozen more that will trickle out to the, to the global population in the ensuing years.
Thanks, Dr. Rozhon. I have some related questions. Once a drug is approved, how quickly can it be distributed globally?
Well, drugs are different. So the pharmaceutical companies, once they submit their marketing application, if they're pretty satisfied with the results. In other words, one of the things you want to see in your phase 3 clinical trials for sure is that there is benefit from the drug, and the benefit of taking that drug outweighs the risks. And when we talk about risks, we're talking about the side effects. So if your phase 3 clinical data show unequivocally that patients benefit and the drug toxicities are not going to be, terribly significant, then almost certainly you're going to get FDA approval. So if companies have been able to generate those kinds of results, companies will start their manufacturing process as soon as they submit the marketing application. So generally speaking, by the time FDA gets around to approving the drug, which, depending on, a drug, for example, that would treat COVID-19, I think you could expect FDA is going to work really quickly to get that. So but companies will be ready to ship the day they receive the approval. How much they will ship depends on how much they've been able to manufacture. But companies usually gear up quite quickly within the first year of approval to have sufficient newly approved drug out to the populations who need it. Now, given, of course, we're talking about a disease that is very extensive across the globe right now, there may be some delay in getting it out, but I'm sure the company is going to work as quickly as they can because, for many people, this is a life and death situation.
One question is, earlier you mentioned that a previous disease had burnt itself out. How does a disease burn itself out, and can this one?
Yeah, like SARS-1. I did use that terminology. So I'm not sure I understand how it burned itself out. I'm not sure that I've seen anything in the literature that explain that, except that in the countries where it was located, they had pretty good contract tracing, started in China, some other Southeast Asian countries, and the Chinese in particular and some of those other countries basically were able to identify those infected and then quarantine them so they could not spread the virus. Now, the other thing about SARS-1 is that there doesn't seem to be the reality that people are highly able to transmit pre-symptomatic, and there's no evidence that people there wasn't any evidence of asymptomatic people. So if there was no evidence of asymptomatic people, then you wouldn't have that transmission. That explains why SARS-2 is so much worse than SARS-1. But I think in part it was a different virus. Didn't seem to have the same degree of transmissibility, and the countries where it first appeared, the local health authorities took pretty aggressive steps to isolate those people. In fact, there were 2 or 3 patients who ended up in the United States, but they were also identified quite quickly. I think one was in San Francisco, the other was LA, coming over in flights from China. But basically, the disease never got a foothold to take off in the United States then. And I think you have to give a lot of credit to the local health authorities who identified it and quarantined those patients, both the small number here as well as the larger number of patients, a couple of thousand in Asia.
Excellent, thank you. Another question is, Estemara is Estemara good for treating COVID-19, and why has there been so much focus on hydrochloroquine?
Yeah, so the Actemra, I think, is the molecule you're talking about. So this is a monoclonal antibody that targets IL-6. So the focus on this is that it dampens down the inflammatory response. So one of the things you've probably read about people who have serious disease, those that are often on the mechanical ventilation, have really what they call a cytokine storm. Basically, this is an inflammatory storm where the body is really overreacting to the infection. And so by the Actemra antibody will target IL-6 and neutralize it. So this has a pretty significant effect in dampening down the inflammatory response. So, but beyond that, there's also data that's come out quite recently of another drug, and that's dexamethasone. It's a corticosteroid drug that also dampens down the immune response, and it's already approved for in the United States and other countries. So it can be used immediately to dampen down the immune response, and the evidence so far is kind of trickling out, but it does suggest that once you dampen down that inflammatory storm or cytokine storm, then you can actually end up with patients who are more likely to survive the more severe infections.
Thank you. Are there different variations of the virus, and why can some people get it twice?
Yeah, so there are lots of different variations of the virus. There's several thousand isolates, and none are genetically identical, but they do have a high degree of genetic similarity. And so the key things when you think about vaccines are developing vaccines against a protein that could mutate and vary. that would of course diminish the functionality of the vaccine. So it turns out that SARS virus is an RNA virus and it can mutate. But interestingly, unlike HIV or unlike flu, it has a system of monitoring for mutations and can actually correct those. So in fact, the SARS-2 virus genetically has a gene size of around 30,000 nucleotides. So in fact, this makes it one of the largest, if not the largest, RNA viruses. So meaning it has more genes to do things that other viruses, RNA viruses, don't have. And so for whatever reason, through evolution, it has evolved a process like human DNA and mammalian DNA to identify mutations and actually fix them. So in fact, the overall mutation rate for SARS is something like 2 nucleotides per month. And so far, that doesn't seem to be enough to cause us any problems in a virus escaping any of the vaccines that would be in development. So I think that's good news for us, but I think it's something we'll still have to monitor to see over time. But I think so far it looks like we won't be in a situation where we would, assuming the vaccine produce antibodies and immune response that will last for a long time, we wouldn't be in the situation where we have no vaccine for HIV and a vaccine for flu that we have to re-administer every year. I'm sorry, the second part of the question was?
Why do some people get it twice?
Oh, so so first of all, we know with the SARS viruses that cause the common cold, the coronaviruses that cause the common cold, is that they produce antibodies that are short-lived. They seem to be gone in 2 to 3 or 4 months. So that explains why they can get reinfected. The data coming out on patients who have the COVID-19 disease, it's anecdotal that they can get reinfected. It may be also possible that for some reason they have not cleared the original infection. Maybe they've knocked it down. So the answer to that question is we don't know. So more research is needed to actually provide data to nail down that question. So first of all, I don't know that it's true that people who have COVID-19 actually can get it again. As I say, there's anecdotal information out there that is the case, but some of those people have been shown to be, they didn't clear the original infection. So I think time will be needed and more data, more study to fully resolve that issue.
Excellent, thank you. There is another question. Could you talk a little bit about the major similarities and differences between COVID-19 and the Spanish flu? I'm not sure if there's a lot of time to get into that.
Yeah, well, let me say, Spanish flu was influenza. So it was a very significant genetic change that occurred in that virus. So flu has a segmented genome. So they have 8 different segments of their genome. And if 2 different viruses infect, say, the same animal like a pig and the same cell, those 2 different viruses can actually reassort their genetic material. So what can come out of that would be a virus that could be quite different in terms of its pathogenicity for humans. And so basically that particular virus at the end of World War I was very pathogenic, easily transmitted. And of course, those were in the days where we didn't really know much about viral infections. We didn't really know how to treat them. We didn't have a lot of the modern equipment and understanding in medicine that we have now. Anyway, the long and the short of it is, it's estimated that the Spanish flu ended up killing like 50 to 100 million people, between 1918 and 1920. So different viruses, different times, a different scenario. So I don't think, because it's likely that we would ever have to have that concern about COVID-19, because one, it doesn't have a segmented genome. If there are mutations that are occurring, it's at a low rate. So what you might find is the possibility of a gradual increase in its virulence capabilities, but it's not going to happen overnight. That happened like with flu, because they're completely different viruses. And again, flu has a very different genome, a segmented genome.
Thanks, Dr. Rozhon. I think that's really nice to hear, because everyone's comparing the two. So
yeah. I mean, the other thing, there's lots of differences about flu in that, it seems to be more seasonal. It kind of burns out in the summer. and then comes back in the seasons where, you know, it's cold and people tend to stay more in house. Whereas, you know, there was a lot of talk about COVID-19 being seasonal, but I certainly think we're in the warmest month of the years right now, and we're seeing tremendous spread. So I think the conclusion is that the SARS-2 is not a seasonal virus.
Mhm.
So there are some really major differences. So I wouldn't spend, I mean, they both can cause severe respiratory infection and fatality, but beyond that, the SARS-2 virus is quite a different virus in terms of, you know, total number of mortality, severity of disease, and really the greater number with SARS of the organ systems it can impact compared to flu.
Thanks, Dr. Rozhon. I hope I'm asking this question correctly, but are viruses inside a cell motile, or do they just float around randomly until they get something for the next step?
They float around randomly until they find something for the next step. No, they don't have any mechanism of you know, of really being mobile within a cell, you know, other than diffusion and Brownian motion. And, you know, I didn't mention it, but when a single virus infects a cell, and again, it varies from virus to virus, when a single virus infects a cell, it can produce thousands of progeny of virus. And I've not actually heard what the numbers are for SARS-2, but there's so many viral components and elements, enzymes, structural proteins flowing around. They don't seem to have any problem finding what they need to mature into viral particles that, you know, can get out of the cell. But as far as we know, there's no like avenues or routes that these viruses take within the cell, other than just sort of randomly bumping into enzymes and other cellular elements that are necessary to propagate and mature viral particles.
Excellent, thank you. Let's see, what areas of virology will be most in demand in the coming years and decades? You did go over that a little bit.
Certainly, there's going to be continued tremendous research in, you know, for antiviral drugs and for better vaccines for, you know, for SARS-2 and COVID-19. You know, we still have SARS-1 is circulating. We still have MERS circulating. We still have a number of respiratory viruses. I mean, we can even do better in flu. Why can't we come up with a vaccine that would be that would not have to be given annually? So there's still many questions. There's, you know, viruses that are thought to be involved in human cancers. So there's tons and tons of areas of research yet to be done in virology. And you know, if you look at the track record of newly appearing viruses in the last 20 years, so what we've had SARS-1, we've had MERS, we've had Zika, we've had Ebola. So, you know, virology is going to keep busy because you know, new viruses continue emerging, and I think we're going to see more and more of that as we go forward into the future.
Excellent. Can you recommend a reference for viral particle structure and SNME proteins?
You know, there's lots of references. You could go to Wikipedia and just type in, you know, SARS-CoV-2, and you get everything you probably would want to know about the structure and the non-structural proteins, the viral replicative process. If you're interested in more deeper scientific detail, you could go to Google Scholar, which is more scientifically oriented, and do a research there. But, you know, if right now you type anything in about SARS or SARS-CoV-2 or COVID and punch search, I mean, you're going to end up with thousands and thousands of articles based on really what's been published in the last several months. It's just absolutely incredible. As I said, I've never seen anything like it. So I would start just, you know, very basic and go into Wikipedia and search basically everything in Wikipedia that they have to tell you about SARS-CoV-2 and COVID-19. And I think you'll find quite a lot of information there. I've looked at it myself quite recently, and it's all really accurate, up to date and accurate.
Thank you. This is like a lengthy question. Moderna, Pfizer, Oxford are beginning phase 3 trials. Data may be available by Thanksgiving. How long is it likely to take the FDA to approve a successful vaccine? Will it be shorter period of time than usual for a vaccine? And what effect does policy have on this?
Well, politics, I'm not going to say, I'm not going to say
I
policy.
know.
I'm going to say politics should not have any impact. I mean, obviously the scientists at FDA, they're under the gun, they're under pressure. We know this, but they're not going to cut corners and allow a vaccine to be approved that is not safe. So they're just not, and I have full confidence in FDA and the government scientists working on this, despite what pressure they come under from the administration or that kind of thing. So the thing is, it would be a disaster to the company if one of our regulatory authorities released a vaccine that was unsafe. So I just can't envision it happening. And FDA has stuck up for itself, and they removed the emergency use authorization for the malarial drugs, the chloroquine. So I think they're making the right decisions, and I have faith they'll make the right decisions when we do get a vaccine out there. I would be, as I said, more concerned about how good the vaccine is in actually producing a long-term immunity.
Thank you. Why do you think phase 3 studies are planned to occur in the UK, Brazil, and South Africa?
I don't have any idea that the companies, they may have subsidiaries there. I do not know specifically why they selected those countries. I really don't have an answer to that.
Okay. What are your thoughts on studying the efficacy of Chinese medicine herbs for treating COVID-19?
Yeah, it's a good question. I would say, one thing is I haven't heard much from from Chinese scientists saying those have much activity against COVID-19. So I guess one of the things to answer that question adequately, I'd have to know, are there any herbal or botanical extracts that have provided any data showing that they have activity against SARS-2. So if the answer to those questions were yes, then I might be inclined to say, well, maybe they should be explored. There may be a pharmacologically active substance in an herb or a plant extract that could have activity against against the disease, against the virus. But the first thing I'd want to know, are there such extracts or plant derivatives that exist?
Absolutely. So perhaps the data is just not there.
Well, it might come, but I don't think, I think you'd want to see some evidence for that before you'd want to engage a scientific group on that scientific question.
Thank you. And then do you know what is the latest in terms of virus survival on surfaces and risk of transmission?
Yeah, well, I kind of think the data I've seen, plastic, cardboard, paper, metal, a few hours to a few days, I still kind of think, whenever I go out, I'm, to shop, like food store, drugstore or something, I always wear gloves. And even when when I do shopping, I wash the gloves first with disinfectant before I even take them off, and then I wash my hands carefully. So I would be, when you go out into public places where people touch a lot of things, I would expect there to be virus on fomites, objects. And so that's why, I wear gloves and I use hand disinfectant on the gloves first. That's why I also mask up when I go out. This country is kind of challenged right now in masks and personal protective equipment and so forth. But the bottom line, if we're going to get out of this, this national forest fire we have with this virus spreading, the only way we're going to do it is stopping transmission. And, you've got to, if you get sick, you've got to isolate. If you go out unmasked and don't pay close attention to where you are, or if there are lots of people around, you're going to be, your luck will run out sooner or later. I mean, that seems to be the rule. So to me, what I do is I assume everybody I meet outside my own family house is potentially infected with the virus. So I take the utmost precautions to minimize my interactions with those kinds of individuals. So, I mean, the only way we're going to stop this is for people who are infected to not be around other people. We've all got to isolate and stay away, certainly for people we don't know. But I know it's hard. It's really, really difficult. We're all feeling it. So,
in your opinion, are the SARS-2 strains different enough to cause different symptoms or mortalities? And would different vaccines be needed for different strains?
Well, no, I think I've answered that. What I'll say is I don't think the different isolates are different enough to merit different vaccines or even different drugs once they come. So, I mean, there are subtle differences, but in the key drug targets and in the key viral antigens that are being currently targeted for vaccine development, they're really kind of focusing on those parts of the virus that cannot afford too much change. So they have to be, the nucleotide sequence has to be retained, because otherwise you'll end up making a mutant protein that won't allow the virus to, for example, to get inside a cell. So I think what we have in terms of drugs in the works and vaccines in the works will be applicable for all of the isolates of the SARS-2 virus that we have circulating around now.
Thanks so much, Dr. Rozhon. So I think I'll just end with this last question, and that is, what preventions can we take to preempt further pandemics?
Yeah, so that's a really interesting question, and I made a little slide on this. Obviously, if you look at what's happened in the 21st century, with SARS-1 and then MERS and then Ebola and then Zika and now SARS-2, it looks like we are on a path for new infectious agents to keep coming out. And I kind of think this is what we're likely to see. So I don't think, as my first sentence says, we can probably prevent it, but there are steps we can take to minimize the impact on society. So first, we need to have a well-funded and fully staffed global infectious disease surveillance network. We have one of those already in place for flu. So one of the things the World Health Organization does on a yearly basis is they have stations throughout the world that are monitoring for new flu viruses. And based on what they see in those global stations, that actually dictates what the global pharmaceutical companies will do in terms of generating the vaccine for the upcoming year. So if we had something like that for emerging viruses or other infectious agents, that would give us an early kind of heads up that we're seeing something new. So that basically could then trigger a lot of other things that could come into play quickly. So in the event of a pandemic, the response must be led at the national level rather than the variety of responses we've seen in the States. So the countries that have done really well in controlling the pandemic, the number of people infected and deaths, have been companies that have had really nationally led programs. And so every province or state or whatever is following the same set of rules. In the United States, we've got a plethora of all sorts of different openings, closings, government. It's been a disaster and will continue to be a disaster, I think, until we get a nationally led program where everybody is following the same standards for controlling this disease. The evidence is pretty obvious. Those countries that have done that are the countries that have the lowest numbers of infections and deaths. So certainly, really, that's something we need to do. The other thing, of course, is we really need to rejoin the World Health Organization. So the U.S. has had a global leadership role in fighting all sorts of these emerging diseases for virtually ever, and now to withdraw and be away from where all the action is globally. We can't do that. We need to be involved with everybody else who's fighting this. So we probably need, in countries where they have these wet meat markets in Asia and in Africa, people going out hunting wild animals and bringing bush meat back into cities for sale, probably not a good idea. You could be introducing foreign viruses or animal viruses into human society. They make the jump to humans, and you're on your way to an epidemic or pandemic. So a lot of people are building now and interfacing in wilderness areas, again, places close to wild animals. Probably not a good idea. So invest in and maintain staffing in national health organizations. So US CDC, I mean, for some reason now in the US, it's mostly the politicians that are leading this attack on SARS-2. It shouldn't be. They don't have the expertise to do it. Should be the CDC, but the CDC has been sidelined for whatever reasons. So really, it should be the scientific medical community leading. So yes, they're going to collaborate with politicians. I mean, there has to be a balance between how we try to control the disease and to what extent we close up society. I mean, obviously, terrible situation for people who've lost jobs and so on. So there has to be a balance, but the driving decisions for how to get rid of the virus has to come from the scientists, because the politicians do not have the expertise to do that. We need to certainly develop and maintain local contract tracing programs and personnel, have them at the ready when, you know, the first flag is raised, there may be a new pandemic. The leadership of the CDC and the state health departments need to be professional people, not political appointees. This has never worked well in a society where the chief people leading scientists are political people as opposed to scientists. That's just not going to work, and the United States has demonstrated very nicely why that doesn't work. The U.S. government needs to invest more money in emerging disease research and enhance the readiness of platforms for rapid development of new drugs and vaccines. So you might be wondering why the Oxford vaccine has been so quickly developed. Basically, they had a platform for other vaccines they were developing with their chimpanzee vectored adenovirus. So they just simply plugged in the SARS-2 spike protein into that system to make their vaccine. So they could do that, you know, as soon as they had the sequence for the vaccine. So if you have the platforms for drug discovery set up to go for viral diseases and vaccines ready to go, when a new virus appears on the scene, then you can be ready to push the effort for new drugs and vaccines really quite quickly. And finally, I think a very important point is limiting the impact of future pandemic. It can't be an endeavor on a single country. Really, it must be a global effort. No single country can be safe from, you know, a virus like SARS unless all countries are safe. So to have the United States out of the World Health Organization right now is the worst decision that could have been made in terms of protecting this country from this current virus. I don't think we can do this alone. So that's why my personal feeling is, from the scientific perspective, everybody has to be within the same system and base in terms of how they're attacking this virus, because if you don't do it right, then it's going to get out and infect more people. And the more people that get infected, the more spread you have. We see it every day. I mean, we've passed 150,000 deaths in the United States this very day. So no suggestion right now that it's going to stop. So it concerns me. So this is what I think we need to do. Some things to prevent future pandemics. And I know some of it seems kind of extreme. Maybe some of it seems political. But we ought to at least be talking about what we have learned from the current pandemic. So for heaven's sakes, what we have learned and we could apply to get out of the current problem, and then to apply that learning experience, that knowledge, to what we would do the next time we see a potential pandemic on the horizon.
Thank you so much, Dr. Rozhon.
My pleasure. Thank you.
Thank you. So we did get a couple of the questions we didn't get a chance to go over, but we've gone much over the webinar time. So I'll send those questions to you separately.
Okay.
And we've also gotten a lot of comments just thanking you for the discussion and your opinion. A lot are coming in right now. Totally agree with you, Dr. Rozhon. Great presentation. Thank you. So I'll send all that to you later.
Okay. Thank you.
And then could you actually show the last slide? So again, thank you everyone for attending. And Dr. Rozhon, again, thank you so much for this presentation.
My pleasure.
Everyone, I have linked his instructor page in the chat box. So if you want to learn more about Dr. Rozhon and connect with him on LinkedIn, the link is in the chat box. And then he does have 2 upcoming courses. So feel free to take a look at those. There's going to be virus vaccines and antiviral therapy class that starts Monday, August 17th. So take a look at that. And UCSC Extension does have a 25% off summer discount, which is not often. So take advantage of that if you can. And then for those of you who are interested in more content, we have webinars every week. Next week, we'll have Elizabeth Eastman talking about the topic of teaching adults using a trauma-informed wellness approach. And if you're also someone in between jobs or looking at a career change, then we have Lee Pham, who will be talking to us about marketing yourself. If you don't do it, then who will? Again, thank you everyone so much. And Dr. Rozhon, thank you for your time.
And thank you for your questions. I really appreciate it. All right. Take care, everybody. Stay safe.